and this summer I was given the opportunity to work in the non-lab, and my topic is understanding the impact of long-term and short-term R1881 treatment of LNCAP cells. Okay, so let's start with the basics. Prostate cancer is an uncontrolled growth and spread of abnormal cells in the prostate. This abnormal growth is called a tumor. A tumor in a non-vital space is a benign tumor, or a tumor that needs to be treated but is not life-threatening. And cancer on a vital organ or that metastasizes to a vital organ is a malignant tumor. Cancer can also metastasize or spread to other parts of the body. And for prostate cancer, these places are often the bone or the lung. And to zone in on my topic, let's talk about androgens. Androgens are basically sex hormones, and in men, the most prevalent androgen is testosterone, and in women, the most prevalent androgen is estrogen. To provide some more background on my topic, LNCAP are cells that are derived from the lymph nodes, which is a part of your immune system, and from a Caucasian man with metastatic prostate cancer. These are the cells I used in my experiment. R1881 is a synthetic androgen receptor agonist. It is structured similarly to dihydrotestosterone, or DHT. However, R1881 is a lot more stable, so it's easier to use. And then HER3 stands for Human Epidermal Growth Factor Receptor 3. HER3 is a protein that's commonly found in high numbers in men with lethal prostate cancer. HER3 is also correlated to shorter time to castration resistance, meaning the cancer advances faster as well, and it aids in cell proliferation and cell growth. My rationale is that the lab found African American men have higher DHT in the prostate compared to white men. Therefore, our objective is to investigate protein expression resulting from different treatments of R1881, and to sum up my hypothesis, it's basically that androgen signaling is affecting the prostate cancer by making differing levels of HER. And we started with cell culturing, and cell culturing is basically how you grow your cells. And the thing is, there's different ways to grow your cells, and that's what makes it interesting. And most experiments revolve around growing cells, which is cell culturing. And then we used EVOS, which is just a microscope that's computerized and allows you to take pictures and zoom in further than most. And I use that to track my cells' growth. And then this is the big boss, y'all. This is the 48-hour versus 6-month experiment. And basically, you start this by culturing 3 plates until they're around 70% confluence. And these plates contain LNCAP cells that have been adapted over 6 months to either vehicle, 1 nanomolar of R1881, or 10 nanomolars of R1881 media. And then upon reaching ideal confluence, the cells are passaged at 1.4 million for each respective group in the long and short-term treatments. And then the 48-hour treatments have just 1 nanomole of R1881 and 10 nanomoles of R1881 media, while the 6-month had 1 nanomolar of R1881 media, 10 nanomolars, and then a vehicle. And then after 48 hours, the cells were imaged and processed for Western blot. And then we've extracted our proteins. So in protein extraction, the proteins are found in varying different parts of the cell. So after the cells grow for 48 hours, we lysis them and access their available proteins. And then a Western blot is used to detect proteins by denaturing the proteins in water through a 3-step process of separation, transfer, and detection. And moving them to a PBDF membrane, the antibodies are used to bind our proteins of interest. Once we image the membrane, we quantify the protein's band using the image studio to measure for fluorescence. In this experiment, all outcomes are being compared to my vehicle, which is basically cells that went through the same process, but the normal way, or in this case, the way without R1881. When you compare the HER3 expression in the cells with R1881 in them to the vehicle, you can see that in the 48-hour experiment, there was much more expression in the 1 nanomole of R1881 adapted cells that even passed the vehicle and HER3 expression. But in the 6-month, both the 1 nanomolar and 10 nanomolars adapted cells had underexpression of HER3. And then not only did the 48-hour treatment cells express more HER3, but they also grew faster. A common technique to saving the lives of men with prostate cancer is to undergo a prostatectomy. The problem with that is that some forms of prostate cancer are castration resistant. This research would be a biomarker that would keep men from going through an unnecessary prostatectomy if they're castration resistant. So with that, here's my conclusion, and to give you a general overview of it, it's basically you can see through this experiment that HER3 expression is regulated differently with time, and these findings together suggest a potential relationship or correlation between the androgens of HER3, which may relate to the initial point that African American men have higher androgens in the prostate. So with that, here's some different ways we could use this experiment and change it to help different causes. The first being using MDA-PCA2B cells, which are cells derived from the bones of an African American patient with metastatic prostate cancer. And prostate cancer is more aggressive, lethal, and prevalent in African American men and men of Caribbean descent. So this would help lower health disparities between African American men and other races. And then we could also use cells that come from a real patient to help that patient find out what they could do and see if their prostate cancer is castration resistant. Here are my references. And now I'd like to acknowledge some people that made this presentation possible, starting with anyone who's donated money to the UIC chess program, and to the non-lab members for teaching me the ropes and how to work in a lab. And finally, a very special thank you to Larissa Nunn for having me in her lab and making me feel like this is my second home, and to Robert A. Holt for doing crazy things like coming in way earlier than usual, walking me through experiments I don't know how to do, and just helping me dive into this topic I've never really looked into before, and this experiment wouldn't have been the same without you. Any questions?
Chlorination and ozonation treatment for E. coli reduction in contaminated surface water. I did my research under Sam Dorovich and Pamela Rich. So to start off, many techniques coexist in water treatment systems to disinfect surface water sources like lakes, ponds, or rivers, and additionally groundwater sources like underground wells. This includes UV light treatment, ultrafiltration, and even desalination techniques that have all been proven to be highly effective in removing a wide range of contaminants from water. These advances in water disinfection treatment systems, however, have led to increased concern about costs and accessibility issues, especially in communities that, for one, may not have access to these advanced systems, as well as lack of proper regulations for contaminant disposal to keep their drinking water safe. When looking at other treatment methods, like chlorination, which is known for its low cost and effectiveness, this treatment is a primary water disinfection solution in many countries and communities around the world. The main issue with chlorination treatment is the association with carcinogenic disinfection byproducts. Chlorine or bleach in itself is not cancerous, but when come in contact with the organic and inorganic substances found in surface water, many disinfection byproducts can form as a result. The two main ones associated with chlorination treatment are trimethylanes and halixic acids, and many studies have shown a positive correlation between bladder and colorectal cancer with long-term exposure and high intake of these byproducts. Removal for these disinfection byproducts is possible through granulated activation of carbon technology as well as air stripping techniques, but once again, these techniques are very timely and costly, which bring into question how it may be able to be implemented in those developing countries and communities. So this brings into question how alternative treatments compare to chlorination in terms of effectiveness of treatment, as well as safety and limiting the risk of those disinfection byproducts. So what we decided to look at was ozonation treatments, and this involves the use of ozone gas to oxidize a variety of bacteria and contaminants in water. Compared to chlorination treatment, it removes a lot of those taste and odor problems that are consistent in that treatment, as well as limits the do not have any residual disinfection products left over after the treatment like chlorination does. Because ozonation involves a combination of O3 gas and once again the organic and inorganic substances found in surface water, disinfection byproduct is bound to form. But the difference between ozonation disinfection byproducts and chlorination disinfection byproducts is the risk that it has. So the known ones found in ozonation treatments are aldehydes, ketones, and carboxylic acids. But once again, because they're found in very little concentration, the health effects are very limited. Additionally, a lot of these byproducts eventually mineralize later on in treatment and can be filtered out of the water. So while it was really important for us to compare those disinfection byproducts, what we really wanted to look at was the effectiveness of ozonation treatment compared to chlorination treatment. And we did this by looking at the removal effects of a specific contaminant, and we decided to look at E. coli. So we did this by performing chlorination and ozonation treatment on Lake Michigan water samples that we collected. We ran 3 trials for each treatment as well. For ozonation, this was done with the use of a microplasmic ozone generator, which essentially breaks apart oxygen and reformulates it as ozone, and then bubbles it through the solution. Additionally, for chlorination treatments, we added, we followed EPA regulations for how much bleach is needed to add to disinfect the water, and we did this for a total time of 30 minutes for both treatments. And then to analyze that decline in E. coli, we used whole wort detection culturing methods to analyze changes in total coliforms found in the water as well as E. coli. So because of different environmental factors, the initial amount of E. coli found in each trial differed. This was mainly because of, like I said, environmental factors. So before some of our trials, there were a series of thunderstorms that happened, which affected the water quality, which in turn either increased the E. coli count on some days or decreased the E. coli count on some days. But overall, these differences were relatively small, making our results still very comparable. In terms of the effectiveness of both treatments, chlorination and ozonation after 30 minutes both resulted in 100% removal of E. coli. But more specifically, for our ozonation treatments, in trial 1, complete undetection of E. coli occurred between 10 and 15 minutes, and in trial 2 and 3, complete undetection of E. coli occurred between 5 and 10 minutes. For chlorination treatment, however, complete removal of E. coli occurred well before we took the first sample. These results were consistent in both the first and second trials. So for the third trial, we did some additional sampling to see if we could quantify that change more quickly. But even, and we took samples at 10, 20, and 30 seconds. But even then, E. coli was not detectable at 10 seconds, which shows how quickly this treatment worked. So once again, both ozonation and chlorination treatment resulted in 100% elimination of E. coli after 30 minutes, but chlorination treatment worked much faster. Despite how quickly and effectively chlorination treatment worked, disinfection chemicals were still left in the water even after the treatment was completed. Bleach or chlorine eventually does break down in water, but the rate at which that occurs depends on a lot of factors, like the concentration of bleach added, the temperature of the water, or even different environmental factors. But typically in diluted solution, it can take from a few hours up to a few days for the bleach to completely be gone. So this just increases the risk of those disinfection byproducts forming later on after the treatment is complete. Additionally, ozonation for water disinfection is still a relatively new treatment method, and there is so far very little evidence related to long-term health effects due to ozone gas exposure. But unlike chlorine treatment, there is no links to carcinogenic properties. These treatments only address bacteria removal in water, so it is still really important to look at different filtration methods to remove those trace heavy metals and debris found in water. before treatment. But ultimately, more work needs to be done to effectively compare chlorination and ozonation treatment. But this was a baseline study to see how ozonation worked in comparison to chlorination, which we saw was effective. So future work can include maybe even combining these two treatments to see how ozonation works to potentially remove some of those disinfection byproducts. These are my references. Thank you so much to the research and UIC or the CRTEC from our leaders like Sam and Nate and Ellie. An additional thank you to my mentors Sam and Pamela. This research could not be done without you.
Dr. Nelson Chen this summer, and my project was biomarkers and clinical factors for predicting lymph node metastasis and survival of head and neck cancer. So, some background. Head and neck squamous cell carcinoma, or HNSCC, represents a significant portion of all head and neck cancers. It is also the 6th most common malignancy worldwide. Although there have been developments in our understanding of HNSCC, the 5-year survival rate has remained largely unchanged for the past 30 years. This is due to its aggressive nature and complex clinical management due to its likelihood of local invasion and lymph node metastasis. Lymph node metastasis is directly responsible for the poor prognosis of HNSCC, with the 5-year survival rate of single ipsilateral LNM being less than 50% and bilateral LNM being less than 25%. Therefore, identifying reliable predictors of lymph node metastasis and overall survival is crucial for improving patient outcomes. So, some more background. Studies have shown that HPV E6 and E7 oncoproteins enhance the activation of epidermal growth factor receptor, or EGFR. EGFR transcribes genes that are responsible for cell proliferation and survival, and it contributes to the aggressive nature of these cancer cells. Although EGFR is correlated with HPV-positive HNSCC, overexpression of EGFR is found in 80 to 90% of all HNSCC specimens. The activation of EGFR also promotes epithelial to mesenchymal transition, which is a crucial phenomenon in cancer metastasis, where epithelial cells gain mesenchymal characteristics and it increases their motility and invasiveness. So, studies have shown that 2 other biomarkers, being Mementin and E-cadherin, also play a role in EMT and cancer metastasis. Lower E-cadherin levels are associated with more aggressive tumor phenotypes and poor overall survival, and E-cadherin is a tumor suppressor. Mementin is a marker of mesenchymal cells, and it's upregulated in EMT, and it promotes increased motility and invasiveness. So, although these biomarkers are all responsible for LNM and cancer metastasis, it is difficult to predict LNM with high specificity and sensitivity using only one of these protein markers. Therefore, our goal was to use statistical analysis methods to identify a relationship between the biomarkers and their covariates to generate a predictive model for overall survival and progression-free survival in order to better predict lymph node metastasis in HNSCC. So, for our methods, we obtained clinical samples from this study from patients diagnosed with HNSCC, then we used quantum dot-based immunohistofluorescence to quantify the biomarkers for statistical analysis. Quantum dots are nanoscale particles made from inorganic semiconductors, and they are more objective and sensitive compared to traditional immunohistochemistry. Therefore, they are the ideal probes for quantifying multiple biomarkers simultaneously, as we did in this study. For statistical analysis, we compared the biomarkers and the covariates between patients with and without lymph node metastasis to see if their effect on LNM was significant. Then, we used logistic regression on each biomarker to analyze their effect on LNM, as well as receiver operating characteristic curves, or ROC curve analysis, in order to determine each biomarker's ability to predict LNM. For survival analysis, we dichotomized the biomarkers by their median, so we have high level and low level of the biomarkers, and then we analyzed progression-free survival, which is time from study enrollment to disease progression, death, or last contact date, whichever comes first, and then overall survival, which is time from study enrollment to death or last contact date. So for our results, chi-square tests showed that radiation, chemotherapy, site, tumor stage, and grade all had a significant effect on lymph node metastasis. And with t-test, we showed that all 3 biomarkers also had a significant effect on lymph node metastasis. For our ROC analysis, our single biomarker models, EGFR had the best predictive power, shown here in blue, And for our multiple biomarker models, the 3 biomarker model of E-cadherin plus Vimentin plus EGFR had the best predictive power overall and the highest the best AUC. As you can see on the table, the 3 biomarker model had the highest area under the curve closest to 1, with 1 being a perfect model. And chi-square test shows that all the area under the curves are different from 0.5, with 0.5 being a random guess. For our overall survival analysis, we showed that E-cadherin and EGFR both had a significant effect on overall survival, with patients that had high E-cadherin having significantly longer survival, and patients with high EGFR having significantly shorter survival. Looking at the hazard ratios, we see that patients with high E-cadherin had around a 50% lower risk of death compared to patients with low E-cadherin. Patients with high Vimentin had around a 39% higher risk of death, although that's not significant. And patients with high EGFR had around twice the risk of death compared to patients with low EGFR. For progression-free survival, we saw very similar results, with patients with high E-cadherin having significantly longer progression-free survival, and patients with higher EGFR having significantly lower progression-free survival. And with the hazard ratios still very similar, patients with high E-cadherin around a 48% lower risk of disease progression, patients with high Vimentin around a 20% higher risk of disease progression, and patients with high EGFR with around a 83% higher risk of disease progression. We also saw that other factors, such as metastasis, had a significant effect on survival. For example, in progression-free survival, patients with metastasis had around 3 times the risk of disease progression compared to patients without. For our multivariable analysis, we want to see the effect of biomarkers combined with the covariates on their effect on survival. We saw very similar results to univariate analysis with the biomarkers, E-cadherin and EGFR being significantly associated with overall survival, metastasis as well. And we also saw that factors such as age also had an effect on survival, with older patients 60 years and older, having significantly shorter overall survival and progression-free survival compared to patients younger than 60 years old. So what is the significance? So currently there's no standardized and consistent method for predicting lymph node metastasis in HNSCC. In the past decade, there have been studies using nucleic acid-based strategies such as RT-PCR in order to predict LNM. However, there are 2 major drawbacks to these studies. First of which being that studying mRNA does not further our understanding of the proteins that are directly responsible for lymph node metastasis. And secondly, due to the heterogeneity of HNSCC, there is a large number of genes that is needed to predict LNM, which can be hard to validate. And that can lower the accuracy of these predictions. So using protein-based and personalized molecular predictors can help us achieve this accuracy that we need. So in conclusion, we found the 3 biomarkers in our tumor samples. All 3 biomarkers have significant predictive power for predicting LNM. However, we found that the combination of EGFR and E-cadherin demonstrated the strongest predictive power overall. We also showed that using these multiplex biomarkers based on quantum dot technology can offer a more accurate prediction and better prognosis compared to using single biomarkers. Finally, further validation of our model is warranted in large-scale studies, and hopefully the combination of biomarkers and clinical factors can lead to the creation of a standardized way of predicting lymph node metastasis that can be used in a clinical setting to help guide patient treatment and improve and prolong the survival of patients diagnosed with LNM. These are my references and acknowledgements. This project was completed, and I would like to thank Research Star for providing me with this opportunity, as well as the CRTEC team and my mentor Nelson Chen for their help throughout the project. Thank you.
I'm the auditor of triclosan 8 to develop other anti-cancer candidates via computer-aided drug design. Okay, so a little bit of background before we dive in. Okay, so drug discovery is the process of identifying potential new medication that can improve patient lives. It involves many scientific disciplines, both including biology, chemistry, and pharmacology. In the field of drug development, molecular imaging is used to track the activity of a candidate and visualize its interactions with specific molecules and biological pathways that are found within the body. Some traditional methods of molecular imaging include X-ray crystallography and nuclear magnetic resonance spectroscopy. And the process of drug discovery is divided into 5 stages, where we have number 1 being discovery and development, where researchers evaluate thousands of compounds to identify potential candidates for development. And number 2, we have preclinical research, and this is used predominantly in animal models. And at 3, we have clinical research, where we have clinical trials that are conducted on volunteers to answer specific questions about the drug's safety and efficiency. At 4, we have FDA review. So the sponsor submits a new drug application to the FDA to review and approval. And then at 5, we have FDA post-market safety monitoring. So I wanted to include this graphic because it gives you guys a visualization of the compounds that we start off with, which is over 20,000. And you would think that because we start with such a broad number and narrowing it down to just one singular approved drug, this would lead to an upward trend in drug discovery, which unfortunately is not correct. We have a 90 to 95% failure rate in drug discovery, which is why we have adhered to CAD, computer-aided drug design. And here I just want to highlight the process that the compounds undergo. So first we have our compounds, which is just characterized compound. And then we move on, and it develops into a hit. And then we generate it into a lead. And then the lead is then optimized into a clinical candidate. Okay. So some of the benefits of CAD. CAD allows researchers to prioritize the best compounds for synthesis, testing, and progression. So basically with CAD, we are more faster, more efficient with drug design, save us money. We better understand the process of drug development and its receptors, and we forecast computational projects. A little bit more background. So CAD is broken down into 2 approaches. You could either take a structure-based drug design, SBDD, or ligand-based drug design, LBDD. In my research, however, we incorporated SBDD. So we took molecular docking. So our objective. So my objective this summer was, our goal in this research was to determine other possible HDC11 inhibitors that could mimic the therapeutic effects exhibited from TSA. And what that means is we're going to take our target protein, being the HDC11, and that's a zinc-dependent enzyme, and the protein coding gene that is part of the histone deacetylase family. And our TSA is not only a powerful inhibitor in tumor growth in human breast cancer cells, but it also arrests the cell cycle in G1 and G2 phases and induces cell differentiation. Now, how did I go about this process? So the software that was used to perform the protein ligand docking was Maestro because of its user-friendly interface. So from the get-go, we need to make sure that we understand all of the functionalities of Maestro in order to optimize our results, right? So that's the first process. And after that, we have to make sure we have readily at hand our identification code of the molecular models that are going to be used. And the process that this undergoes is, first we identify our target protein, we prepare the protein structure for docking, and then we prepare our ligand structure, we generate receptor grid, and then the docking of the ligand, analyze the dock poses, and then we have a visualization of our protein ligand complexes. Our results. So here we can see our ligand, the TSA, and its various receptors. The reason why I wanted to include this is because, as you can see, there's different ways that this ligand can bind to our target protein, but that doesn't mean that it's going to be the most optimal way of binding, right? Because we could have introduced therapeutic effects, but we could also have side effects, and those are going to be unwanted, right? Next. Now we can see our ligand in its optimal docked position. So this is where we saw that after all the other poses that were generated using Maestro, this was the one that produced the most therapeutic effect. And the reason why is we have So it looks like this, and we basically constructed a table, which I will show, that shows why this is the best pose, and it's because it exhibits drug-like properties. So this is basically what's going on in the ligand and the target protein. Our TSA chelates the zinc ions in the active site of histone deacetylase. And then here's a 2D interaction diagram of our target protein and the compound, which is my favorite because we can see the residues are annotated with a 3-letter amino acid code and its corresponding chain, and colored as follows. So we have our hydrophobic is green, our polar uncharged is light blue, the negatively charged is red, and the positively charged is dark blue. And then our hydrogen bonds are indicated with pink arrow. So here we have the bioavailability radar, and what this is, it has 6 parameters: lipophilicity, size, polarity, insolubility, unsaturation, and flexibility. So essentially, of the tested compounds, this is represented by a red line integrated into the pink area. Molecules that fall within the pink region of the radar are considered to be drug-like. TSA exhibited compliance to all 6 of these rules. Okay. So the previous radar was based on these properties, and for pentaid, I'm going to be highlighting just one parameter from each section. So the log Kp value, or skin penetration coefficient, indicates the compound's ability to penetrate the skin and cause of toxicity. Next, we have the bioavailability score, and as we can see, it has a bioavailability score of 0.85, which is great, because we want to make sure that it's greater than 0.5, because that indicates its drug-like ability. Next, we have synthetic accessibility. A low score estimates how easy it is to synthesize these drug-like molecules. The score ranges from 1 to 10, 1 being easy to make and 10 being very difficult. Lower assay scores are predicted to be more feasible compounds because they are less complex. Next, we have TPSA, which is sometimes referred to as polar surface area. This is a value that can be calculated for a drug that provides an indication of the drug's polarity and thus an indication of its lipid solubility. Next, we have lipophilicity, the ability of a chemical compound to dissolve in fats, oils, and lipids, and nonpolar solvents. Next, we have log S as a predictor of the compound's negative log of water solubility, which is the decimal log of its molar solubility in water. Conclusion. So based on structural similarities and behavioral properties, we consider these compounds to be generated and optimized into a clinical candidate. So based on our research that we did on TSA, these were the other possible compounds that we derived from that. And what we would do in the future is take each one and go within the same process that we did with TSA. Conclusion. These results test and confirm the findings found on TSA, making this drug an excellent resource when developing other molecules that could perform along similar parameters. These are my references. And this project was completed as a part of the Diversity in Cancer Research Program and made possible by funding from the American Cancer Society. And this endeavor would not have been possible without Dr. Wardrop, my cohort members, the faculty, and my family.